Good day, dear colleagues. I'm happy here in this conference to my second presentation, and today I will try to engage you in computational modeling of atherosclerotic plaque evolution and estimation of mechanical vulnerability of the plaques. Actually, this work developed in the group of researchers here, Mykhailo and Doctor of Medicine Igor Polyvinok, who actually presents the medical center, the Institute of General and Emergency Surgery from National Academy of Medical Science of Ukraine, and I'm here presenting the Polytechnic University. So again, a cooperation between engineering and medicine here. So that's a brief content, a little bit about motivation, and then shortly about the ongoing research, because this project and this research are rapidly growing in our institute. We are interested in how to model these atherosclerotic plaques and to estimate the reliability of the vessels, if it's possible to say like this. Okay, a very short motivation. I'm absolutely sure that you are all known and familiar with that cardiovascular disease are one of the leading problems for death in the world. Yeah? Just imagine that there's 17 million deaths per year. That's absolutely dramatic thing. And almost 70% of men and 60% of women over 65 have the problems of every plaques actually. So that's like medical guys joking that if you think that you don't have this problem, probably you are not correctly diagnosed at the moment. So the more precise looking on that will absolutely show you that you have such a problem. And actually 75% of plaque rupture will automatically provide stroke, infarct, heart attack, and some other problems. So that's absolutely important, practical important, in medical practical important thing, and we need to try to solve. So let me a little bit describe what is the plaque. Actually, for me, for an engineering guy, that was a very strange thing. That's a cross section of a human vessel, and here's the lumen. It's like a blood flow inside, yeah. And all surrounding is the different layers of material. There is so-called adventitia, media, and intima. It's a free layer that are around the vessels of human beings. And as a joke, if you use a swearing words, have some alcohol drinks and a lot of smoking, then closer to the Middle Ages, you will have this problem, this yellow one. So actually, that is in a place there some chemical elements are diffused inside of arterial wall. Yeah, that's actually what is an atherosclerotic disease. Yeah? And here could be so-called this like in a white this, but it could be like a fibrocalcium places, or in some other cases that could be a lipid pool, so-called. And the lipid pools is the most dangerous actually, because they're quite soft. And during the blood pressure inside of the vessel, they could deform, and actually they could rupture due to some conditions. And such a situation and such a plaque called as vulnerable, because it's not stable actually from a mechanical point of view. It could rupture. This cap, this very tiny pink area could rupture, and if it happens, then the absolutely dramatic process of how's in English, thrombotes are rising here, and they could just completely stop the blood flow in minutes, not during the years like the typical plaque growing happens. So we're interested in computational modeling because we're engineers, so we would like to model that. So we propose such a schema. That's a very schematic, and you could see here this blue one is like in the two different layers of the wall, dentita and media, and intima is this purple one. It's a little bit magnified. And the red one is the plaque, which assumed here to be a lipid origin plaque. Yeah? So that's some definitions here. And the interesting, not only how to build this schema, because in the literature there is a lot of study like that, and they typically standardize that, like in a circle, and this, for example, lipid pool as an oval, and then they try to study how the deformation stresses occurs all around here. In our study, we would like to create the systems more patient-specific. So we are going to look on so-called OCT images, this optical coherent tomography images. That's a very specific medical tool. Then some sensor are put inside of the vessel and then make these scanned tomography pictures all around the length of the vessels and also through the thickness of the. You can see this picture magnified. It's a little bit scary actually, but the professional doctors could tell you that, okay, that's more white is a fibrous tissue, more black is a muscle tissue, probably media, and some other is a dentita, which are more soft one. And this circle is actually a sensor that's inside of the area. And if we look very precisely, then some black zones, like this one here, probably a plaque, because it's like deformed the structure, and the lipid plaque absorb all the energy from optical conical sensor, and it will be like in a dark place, yeah, on the picture. So, I'm sorry, that's not in English. We propose to create some software. Today, we even have a computer vision software that could almost automatically do it, but in this presentation I would like to present a manual aspects. Then the doctor could put such dots to demonstrate the boundaries of the main pictures, and then the software semi-automatically to build the schematic model like this one, based on this real patient pictures, and the almost precise information we try to put around this area. The plaque and Artur actually interact between them. So we would like to present this place as accurate as it possible, because actually that is how the plaque could be identified as vulnerable, because it's could rupture. For example, in this place, the medical doctors told me that this is a very, very dangerous place because there's a very thin layer of fiber cap, and it could rupture actually, but they have only intuitive understanding about that, and we are interested in how could we model this. So, yeah, come on. Yeah, that is an information borrowed from the literature about different properties. We use anisotropic mechanical elastic behavior for different layers. That's like a typical normal standard for mechanical analysis. And we're also interested in let me go a little bit forward here. Yeah, that's the model. We build this finite element mesh model and try to simulate how this happens. Yeah? And you could see, like I mentioned here, these places are probably very dangerous here. According to deformation, they have 30%. Actually, for fiber cap, that's more or less okay. It's like not the very risky place, but not so good. And also what interesting we propose to do, we figure out in medical statistics this picture, that is how lipid molecules could be absorbed by the vessel wall during the different years. That's a mistake, that's not a degree, that's a years, 20 years, 46 and so on, and that is this degree of damage. And you can see that's a scatter, and reliability of this data quite huge. Nowadays, in our recent research, we propose to model that as a stochastic process, but in current to this study, we just built by the typical regression model this exponential curve that present the mean value of the growing of the lipid, and we make an assumption that actually this curve, this y, is this exponential, and this is the literature from which we obtain this data set. Allow us to build a different model like a prognosis. So starting from this one, as was taken from real patient, we could prognose how this plaque will grow inside of his vessel during from 20 to 60 years old. And based on that, we built many models and could look on how it's going to be growing. For example, that is like an initial one, the 30%, and here the 40% of strains would happen in this point. So 30% rising of the risk of happenings of some very dangerous situation. And also we try to look on absolutely different situations. Here you could see the possible this collusion degree, so it's how big this atherosclerotic plaque in comparison to the normal cross section of the vessel, and that is different. This range, that is something that could be very easily diagnostic by the medical practitioners. And here is the first principle strain, so the critical this rupture level, and the different bars here indicated by the colors, the different level of the blood pressure inside. For example, we could see it like from normal 120 millimeter on this. How it's pretty.
Pressure of height.
Yeah, and 200 of millimeters of this. I'm just forget the chemical element.
Mercury. Hydrargyl.
Hydrargyl.
It's mercury.
Well,
mercury.
Yeah. Well, liquid metal.
Yeah.
Mercury.
Mercury. Okay, that's millimeter around this mercury level of how the pressure typically measured in
Hg is hydrargyl.
no. Okay.
Okay, never mind. I'm so sorry. I'm mechanical. I like the pressure measured in pascals, but the medical slides some other values. Okay. So the 120 is like something that is okay for NASA measures, and 200 that my brain has. And actually, if we look on everything here, we could say that depends not very easily, linearly, almost linearly. It depends the risk of this rupture. And that is how the stresses, because the process was the strains and the stresses is what actually could go into the rupture. You could see that how dramatically it could be rising, but actually some redistribution of the concentration places occurs, because this is like an initial stage, and you could see that while the plaque growing, actually the level of stress is a little bit reduced. But actually that also could happen. Yeah, but totally they're growing of the plaque. Today, I'm just actually at the finish of all the main results, but today, as an outlook, I would like to say that we are interested in what we are doing now. We model that in stochastic sense. So we try to model the variability of this geometry, because actually this technique, how we identify this line, frankly speaking, is not very nice and very precise, and no one could tell us how to make this better. Actually, there could be a mistake either by human, either by computer vision program. So after that, we proposed that we could identify some line and then assume that it could vary in some region, and using a Monte Carlo simulation, we built not only with one model from this original picture, but 1,000, the different variation of this geometry, and then we built all the strain, all the stresses, what happens here, and we could provide a statistical estimation of what happens with stress and strains for different level of the pressure, and also for different level of the growing, consider all the stochastic process of how lipid could accumulate it in the interval. So that is what we're doing now, and thank you actually for your attention. I think I've finished some conclusions. I think it's quite silly to read them. Everybody's understand them. Thank you very much.
Thank you very much, Elsa. Maybe some questions?
Yeah, I got a question. Now about the application. So here the main idea is, let's say, to have this model developed personally for someone, and then make prognosis for this person, right? So how high the risk is, what the person should do, maybe like immediately go to certain doctors and so on.
Actually, yes, that's the crucial idea, because while the doctors make this optical computed tomography, they just put this sensor and they look on this picture and try to manually to make some understanding what happens. We could provide for them like a software that could say, okay, that's a probability of dangerous place, just like an assistant to help them to make the plan of surgery, because actually if the plaque already exists, the surgery probably will be a good solution, almost always, but because of 75% of the people who actually has such a problem, it's absolutely impossible immediately to plan all the surgeries, and sometimes not only a surgery needed, but some specific like stents could be placed here. That's also a help too much. Now in US they overcome the problem with plaques by putting the artificial stents inside.
So something like mesh or
yes, exactly. This artificial mesh they put inside the vessel, and then it just opens and
like
try to fix the vessel walls from all this problem with deformation. And they do that not only to open that, but to prevent the rupture of the fibrous cap.
And one more thing, when we go back to this like model and reality, like where you had points, and really
this one?
Yeah. What I'm thinking is you need to get data from like either corpses or surgery where they cut open this whole like thing to see what the actual structure is. Then you can see like how well your model is versus the real thing is. Like if you had a real cross section from
unfortunately, that's not like a nice idea. Because there is some study like that. Because if you put this vessel, it's quite small one actually. If you just then put that, and if you just somehow to cut them round in any lanes, they will be like deformed in a new way. And the geometry could be a little bit modified. So we need then the software that could just somehow transfer the new one to the original one or something like that. So that could be quite difficult. There's a lot of study in the literature how they try to investigate the different properties of different vests, and they propose these cross sections and they use them to blow by the pressure from inside, or just to cut somehow into them to stretch that. So a lot of different study, but the deaf tissue and the vivid tissue is also quite different, either through the geometry, either through the properties. But today, these scanners, that's what we have in Ukraine. In Ukraine, we have only 2 such scanners. But nowadays, I think Nokia or some other companies propose a little bit better scanner system that could provide better resolution of this picture, and probably we could just cooperate with that. How to make this better? That's exactly what you told us yesterday about your cells. No one actually knows this actual geometry, and there's a lot of different communication here. Even from human, like Alexei told us, we need to have a better human who could try to.
It's not the case. A cell has exact boundary, and here there is no exact boundary.
Actually,
there is, but we cannot see it.
No, I mean at the picture.
Yeah.
And could you show 19th slide?
19. This one?
Oh no, the next.
This one.
At at normal pressure, we have non-monotonic dependence.
Like oscillations. You mean this big one? Yeah.
What does it mean?
Nice question. I need to think a little bit in more details, not to answer from just directly from this picture. Thank you for another nice question. That could be, I had two answers on that. The first one, positive from the point of model, that's probably the zone of the maximum migrated around the geometry. Or different, that's like what happens, but that should be checked out. And the second one, that could be an error of computations.
But your model doesn't confirm the migration, as I can see here. So
No. Look, the pictures go a little bit different.
okay,
I mean, the distribution a little bit different. And actually the model, this computational model, it will rebuild it, the mesh, the computational mesh will be rebuilt for each particular cases again, and it will not absolutely the same. So the error of integration schema is presented here as well, and the possibility of this migration of this maximum also.
and how do you rebuild the mesh with time?
We have a specific marker software that build the geometry and then to produce the mesh, the parameter of the mesh density as the one integral parameter that is controlled here.
So you deform the initial mesh, or you build a new mesh?
Always a new mesh. For new geometry, always a new mesh.
I mean not geometry, when you build temporal dependence.
What is temporal? For different pressures?
Вы строите эволюцию,
Да.
так? And how do you calculate the next step of this evolution?
In all the steps of the evolution, we build a new geometry and then we build a new mesh.
Okay, how do you change the boundary of this area?
The boundary.
The next boundary.
Based on this, there is some specific algorithm that use this one initial, and then according to how the area should be growing, this area rising.
This is the question. How you calculate the next boundary at the next stage?
All the points, all in our one area and in other one, making an y coordinate step up. The controlling of the scaling up the initial geometry line, considering that the area should be bigger.
So you take the initial boundary and deform it.
Yeah. Yeah. But the original geometrical schema rebuilt after that absolutely like it's in new one. Yeah, it's take this one, then algorithmically built the new line, and then they rebuild everything.
Thank you.
Thank you very much. Maybe some other questions? No. So, thank you, Mr. Lori.
Thank you very much.
You're welcome. So, the next work is the functional variability index, quantitative measure for assessing gene content diversity in microbiology genome. Mr. Drabinsky Nikolai.
Good afternoon to everyone. My name is Nikolai Drabinsky. I represent Technical University of Moldova, and today I will present you our work on genome variability index, a quantitative measure for accessing gene content diversity in microbial genomes. Here you can see that how metagenomic sequencing from hospitals, cities, wastewater and agriculture sites generates enormous amounts of data, and this is our semi-introduction, semi-motivation. it's our challenge. We don't just have a single genome of species. We have we have populations of related bacteria or same species with varying gene content. That's what we call a pangenome. It's a complete gene repertoire of a species. Here I want to show you our motivation. These are real headlines from our region. In 2024, 150 Moldovans died of pneumonia last year, and 6 children died in hospital infected by bacteria. This is why we are motivated to develop tools and methods for pathogen surveillance, characterize bacterial and pathogen diversity. We need better regional and global solutions. Here we can see that this alarming public health situation underscores the critical need for advanced tools to monitor and understand microbial pathogens in our environment. To address this, our research employs urban metagenomics, a powerful approach for directly analyzing the genetic material of entire microbial communities. The following slide outlines the foundational pipeline that makes this analysis possible. From sample collection to sequencing, this process generates the crucial data we need to reconstruct pangenomes and ultimately calculate the pangenome variability index. Thus, this will fall in the essential first step in moving from a clinical problem to a genomic solution. Okay. To analyze microbial diversity, we should to understand pangenomes. A pangenome represents the complete gene repertoire of species or taxonomic group. It consists of two main not models, but components. The main one is core genome. It contains genes shared by all strands essential for survival. The accessory genome includes variable genes found in only some strains, often related to adaptation. Pangenomes range from closed, mostly conserved, to open, highly variable. Okay, we focus on metapangenome reconstruction based on urban metagenomic data, where metagenomic reads from multiple samples are independently assembled and taxonomically deconvoluted to generate spatial level bins. Predicted genes from all assemblies are pooled and dereplicated to form a non-redundant gene set for each species, and here we focus on pathogens. These gene sets are clustered into homologous groups to construct species-specific meta-pangenomes. Okay. This slide outlines the comprehensive workflow for meta-pangenome reconstruction and analysis. This process begins with assembling genomes from metagenomic data and annotating them to identify genes and compute orthologs. Subsequent pangenome analysis generates key outputs for exploring genome composition, gene distribution, and pangenome openness. Okay. Traditionally, pangenomes are characterized using two main approaches. Panels A and B show visual structure. Open pangenomes contain many unique genes, while closed pangenomes are dominated by shared core genes. Panel C shows rarefaction curves. Open pangenomes keep expanding with new genomes, whereas closed ones quickly plateau. D and E depict gene frequency and genomic fluidity. Open pangenomes have many rare genes and high fluidity, while closed ones show uniformity and low diversity. Okay, so now let's introduce our solution, the pangenome variability index. The idea is very intuitive. We measure diversity by looking at how gene frequencies are distributed across genomes. If a gene is missing from all genomes or present in all of them, it doesn't add any variability because there is simply nothing to vary. The highest variability appears when a gene is found in about half of genomes. That's where uncertainty and therefore diversity is greatest. This pattern forms a simple parabola described as a function for f times 1 minus 5 minus f, where f is the gene frequency. Okay. To validate PY, we generated 19 synthetic pangenomes spanning the full spectrum from open to closed. Gene frequency distribution. Yellow curves show open pangenomes, while purple curves show closed pangenomes. Validation using the openness coefficient. The value of alpha smoothly increases from 0.29 to for open pangenomes to nearly 1.0 for closed ones. Okay. Okay, so here we see comparison with alpha and fluidity. If I'm not wrong, it's methods used for describing pangenomes before. Okay. Semi-conclusions. So here we can see two-dimensional landscape. Density of gene frequency and variability across 9 simulated pangenomes with increased core genome fractions. The pangenome variability index provides a frequency-aware framework for quantifying gene content heterogeneity across Mykhailo genomes. Knowing traditional descriptive measures, which often lose resolution at the extreme of genome conservation of divergence, PY remains sensitive across the full openness closure spectrum. This work was made possible by the collaborative efforts of our team: Viorel Munteanu, Alexey Ляху, Dmitry Chorba, Evgeniu Kotlobulga, Nikolai Dravchinsky, Demian Dubchuk, Viktor Yaposkurty, and Viorel Bastan. Thank you for your attention. That's all.
Also, thank you very much. Maybe some questions? Any questions?
What do you, your part of this work?
One more time. What was my part of this work?
Yeah.
My supervisor, I can't say created, but what to say, a new approach. And my part was just as usual technical engineer to create simulations, to make visualizations, to optimize this, because every simulation how to say it, needs more compute power, and not every laptop or PC can run it. So we used HPCs with GPU, with multi-RAM, multi-storage. I created pipelines from input data. Also, I showed workflow. We used R scripts. We used Nextflow pipeline for running step by step all this preprocessing. Yeah.
And how big is this data exactly? Like what size we are talking about?
One more time.
How big the data set size is? What's the volume of data? Just
The metagenomic data.
Curious.
No, one more time.
The dimension of metagenomic data.
Dimensional metagenomic data.
And then multiplied by your number of samples here.
Like gigabytes or whatever.
So one sample is usually near 20, 30 gigabytes. One sample. One sample of metagenomic data. But it can be bigger. So it depends on sequencing diff. The human genome, for example, is 100 gigabytes raw data.
Thanks.
So,
Thank you.
So, thank you very much.
Thank you.
Now,
So, the next work is the special representation of three-dimensional X-ray CT segmentation data using machine learning and mixed reality. Mr. Kasahiro.
okay.
Okay, thank you for your introducing me. And I'm Hiroki Kase from the Faculty of Informatics, Shizuoka University, and Research Institute of Electronics, Shyshkin University. So I'm talking about the spatial representation of three-dimensional X-ray CT segmentation data using machine learning and mixed reality. So first of all, so X-ray CT is used not only the non-destructive testing fields and the security fields, the main using the X-ray CT for medical use. So in the medical fields, so it is used to determine the size of the internal lesions or the damage of our bodies and the tumors in human bodies to diagnose infection disease. So this is this one is spectral X-ray CT is the newest one. So in recent years, so machine learning and deep learning have also been used to examine the CT data obtained not only by the using human eyes but also for analysis and for treatment planning. So using the for analyzing the images. So, but this image shows for the medical professional, for example, the doctors or radiologists, so see these images, X-ray CTs. So this three plane is from this sagittal. So this one is actual images, and this one is sagittal images, and this one is coronal images. So this is good for human understanding, but it is difficult to specify the point to be observed. For example, so we cannot figure out the 3D images of about the damaged part or organ shapes, or where is a tumor or cancer is in our bodies for 3D images. So research is underway to represent data captured by the 3D X-ray CT using the virtual reality called the VR, augmented reality, AR, and the mixed reality, MR. So it's used for the search identification for educational purpose. So medical professor wearing the head-mounted display like this and showing these images on showing on this screen on the head-mounted displays. But so the bad point for using this system is first one is we can see only the shape of the organs or bodies or broad streaming line. But what is important for us to know that inside of the structure of our organs, so not the shape of the organs. And the other one is using head-mounted displays. It is difficult to keeping the cleanness if the medical professor touch the other instrument or touch the head-mounted display. So they have to wash their hands again and wearing the gloves. So and another problem with 3D rendering. So but this is a surface rendering. So other tools. So it's this is a specific. So surface rendering is a way to represent. So selecting a specific result or equality among the pixel value and define it with a desampling function. So we can see the large shading. So and we can know the 3D structure of this one. But there's no density information. So there is no information inside each organs. And shape may not be accurately reproduced between the tissues that are close in the density. So if we settle the threshold line to these organs, so this noise will be shown here, and the manual setting of the threshold and ROI region of interest is required to extract specific organs. So, and segmentation using machine learning and deep learning is proceeding. So, advances in machine learning in deep learning enable complex anatomical structure in the 3D image to be segmented by the organs. And the annotation levels of the organ can be assigned to the each 3D CT size, and coloring and the 3D display are also possible. So we can segment it, so each organ shape of our bodies, and we can see the 3D figure of our bodies. On the other hand, so organs with complex shapes are displayed in an overlapping manner, so the information needs to be organized and displayed. So each of these organs, we can see the colony here, so we cannot see the other organs for these areas. And so the purpose of these studies for the integrally represents the internal structure of the body in 3D space using a mixed reality and machine learning for organ segmentation data. So display not only the arbitrary organs or display cross-section from any direction, in addition to the cross-section at the time of the imaging. So it means the interval of the understanding of the three-dimensional information. So this is the box data as it in the 3D information. So the image of my study is this 3D size cross-section data will be shown to the hairs with using a mixed reality. And if the observer settles this cross-section area, so we can see that not only the 3D surface information, but also we can see the inside information of our bodies. So I use this is a diagram of the representation using mixed reality. So that data taken by the X-ray CT was sent to the high-performance species as the file format called the DICOM. And so using the 3D stress, this is the software, it converts the DICOM data to the surface rendering 3D data. And I used machine learning library called MonaiLabel. So for displaying that in the mixed realities, I used gaming engine called Unities. And this high-performance species is connected to the two devices. One device is the leaf motion controller. This is the motion capture for hand. And another one is the spatial reality display. So this is like a holographic imaging display. So this has a built-in camera, and detects the side of the eye. So this device sends the left and the right and the eye coordinate positions, and this high-performance species outputs the different image depending on the position of the left and the eyes. Yes, and so about the spatial reality display, so these eye position can be detected in a real time in a horizontal, vertical, and depth direction by eye recognition sensor in the spatial reality display. So these eye position and direction are all detected in a real time, and the separate imaging are output from the left and right eyes to achieve the three-dimensional image expression. So these observer feeling the 3D imaging are popping out from these displays. So another device is a leaf motion controller. So this has device requires to moving the subject due to the limited viewing angle is shown in the 3Ds. So this leaf motion controller has 3 infrared LEDs. So let's tackle is infrared LEDs and 2 infrared cameras. So if the observers hover their hand on these devices, so this motion capture catch the movement of our fingers. So the 3D image of our hand will be shown to the screen. So it enables no contact operation by the gesture without using a mouse screen or a mouse or screen touch, and it's capable of taking a picture of the hand and the fingers and locating them in a 3D space by image analysis. So about the cities, X-ray image segmentations, as I used to call the pre-trained model or the mono-level whole body CT segmentation. So
if we compare a non-linear model with different output function, the best quality indicator are of non-linear Gaussian output function. We have best fit of 64. So the obtained result indicators the possibility of improving of quality of this model due the optimal selection of parameters for models. Research of construction mathematical models constructed the basis of real initial data of natural biomedical experiment, demonstrated of high technology effective using MATLAB of surveys for solving the biomedical parameter. Of the next presentation, we see of practical using for this information. The next speech of features of modeling insulin resistance process in MATLAB application package. This for that group. The goals for this work analyze of result of modeling, the effect of process of glucose level of patient. The problem of immunodeficiency as well as ability of control carbohydrate metabolism in the patient body are among the key tasks of modern immunology. We have of a group of 12 patients. We have of two patients of patient B and patient E of under parameter of biomedical control. The step one, selection the modeling of predict of output glucose. We predict of output variable glucose basis on the analysis of polynomial Aries model with a fixed was selected for the patient A and for patient B. The best fit this model of OTA 7 or OTA 5. That very well best fit. Take this modeling, we must control of influence as a parameter. The effect of basal insulin, we prognose insulin for VCs of control result for this dose of basal insulin. For the patient had the little of this is better. The next we see effect of short active insulin. To determine the effect of those of short insulin in patient blood, we think of short insulin of we have result for that patient is patient 1. The short insulin for the 2 The next step we have of carbohydrates. Of carbohydrates, the protected for that, the patient this as a result of carbohydrate input. Okay, we control of this checking of input activity of other patient we control of this parameter monitoring of predict for situation. The other studies of this modeling result of MATLAB package influence of various effects of glucose level in the blood. The patient shows the level depends on this effect, the output parameter, the glucose level on the blood of patient. The observed results show the effectiveness of MATLAB method used, which can contribute the implementation of scientific or practical doc of monitoring insulin resistance of patients with type 2 diabetes, as well as predicting the dynamic of this studied biomedical process for central period of time. Have you a question for this material?
Yeah. So the target parameter was insulin resistance, and which parameters were like the inputs or the parameters that insulin resistance depend on? Heart rate, you said one of them, right? Blood pressure, what else in this model?
Я скорее отвечу по-русски.
Давайте.
Sorry, little English spoke, я spoke of Svenskog for Russia. Если можно еще.
Да, значит, целевой параметр это
что по иксу, что по Y.
Инсулиновая резистентность, а входящие параметры какие? Я видел heart rate.
Входящие параметры у нас использовались, это короткий инсулин, длинный инсулин, использовалось карбогидраты, физическая нагрузка, велотренажер, 30 минут давали нагрузку, и измеряли сердечный ритм. ЧСС. Вот. По этим параметрам построили модель, а потом спрогнозировали появление различных этих самых и посмотрели, как влияет на модель появление этих параметров. То есть мы можем прогнозировать влияние каждого из параметров на результат. Где-то приблизительно за 2,5-3 часа. То есть мы моделируем параметры, моделируем как бы это воздействие и можем знать, какой будет отклик через определенное время.
То есть у вас на входе получается сколько там, 5 или 6 параметров, так?
6 параметров.
И вы прогнозируете вот эту инсулинорезистентность, так?
Да. Во времени. То есть мы получаем, как она изменится, когда у него будет спад, когда будет подъем, и можем прогнозировать эффективность. То есть мы можем, меняя, так сказать, эти значения, выбрать оптимальное значение, которое позволит для данного конкретного пациента, потому что обучение идет по его параметрам, получить оптимальную дозу того или иного.
Понятно. А модель какая в итоге?
Значит, в первом докладе я рассказал, что мы рассмотрели различные виды моделей. Оптимальной оказалась нелинейная модель с гауссовским распределением по выходу.
Так, у тебя сейчас тут как раз точнее. Итак, у нас есть 6 этих самых входных переменных.
Да.
Как они связаны с выходной переменной? То есть там были какие-то полиномиальные модели, я видел?
Мы просто в MATLAB провели по одним и тем же исходным данным построили модели линейные с различными коэффициентами
Это понятно.
и нелинейные.
Вот нелинейные какие?
Нелинейная модель у нас с гауссовским распределением выходной функции.
Хорошо, спасибо.
Еще один момент. Возможно, стоило бы использовать параметры как heart rate variability в качестве входного параметра. Тоже может быть полезным. Но это более информативно, чем просто heart rate.
Да, эти исходные данные, предоставленные Академией Шангельской, они попросили сделать именно, вот, ну, у них был проведен натурный эксперимент, и на этих данных они просили просто обработать эти данные для дальнейшего определения. Сейчас будет сделать более глобальный эксперимент, чтобы уже, используя эту методику, ну, развивать это направление. Спасибо. Так, первую часть мы заканчиваем.
So the first session is already end. So it's 3:30 o'clock. Where will be the coffee break? Okay. So
Our coffee break has been done, and second part of our section starting right now. So let's introduce myself and my presentation. Remote monitoring of human gait parameters during post-traumatic rehabilitation of the musculoskeletal system. This work is a series of work of our team that include me, Andrei Zuev, Roman Tomaszewski, and Alexander Androsov, and we guys research some different parameters of gait, including persons with low limb implants, and now first speech was yesterday that I showed hardware software platform for gait analysis. Now I just shortly explain actuality of this problem. So for now, 445 million individuals with traumatic amputation globally, and this number grows every year, and for now we don't have any official statistics in Ukraine, but non-official statistics give us estimates from 20,000 up to before 50,000 amputations because of war, and this very young men that have amputations of lower limb, and Ukraine maybe counts many as up to 100,000 amputations. So it's a huge number and very big problem. So if describe gold standards of measuring gait parameters, we have two areas, two directions. in this area. So first is using mock-up technology, like on this slide. So there are the cone motion and OptiTrack, but this technology just in lab using and very expensive for using. Alternative way is using IMU-based technology that give us small mobility sensors and give us possibilities measuring parameters during real life in long term. But many researchers don't analyze segmentation and motion different segments of limbs. So it's described only general behavior of patient, of some symmetrical, asymmetrical walking, but not segment like hip of shin, and that don't give us such detailed analysis how person walking. So only just some global parameters. So key motivations for current study, you can see on this slide. There are medical rehabilitation perspectives. So clinical practice and rehabilitation of patients with impaired leg mobility shows that normal human gait is characterized by symmetry in the time patterns of movements between the upper and lower parts of the legs, as well as between the right and left limbs. Second one, importance of pattern identification. In this part, we're detecting deviations from these normal symmetry patterns, provide essential information for quantitative assessment of both the level and the type of dysfunction or distortion in gait. And next one, practical benefits of quantitative gait information. We have enables calibration and fine-tuning of prostheses and other assistive devices, and support visualization of gait irregularities, providing patients and therapists with clear indicators of incorrect movements, which is highly valuable for the rehabilitation process. Final is a technical challenge. To achieve this, it's necessary to develop a multi-sensor wearable system capable of collecting and synchronizing signals from several measurement sources, as well as to design methods to make it possible to obtain generalized information about the quality and characteristics of the gait. Little focus on the last part. So multi-sensor wearable system I showed on the yesterday, and you can explain this on our work that I presented, that was presented yesterday on section, and I described in detail this hardware-software platform. Now I explain methods of measuring and results of this measuring procedures.
short introduced of our hardware-software platform. Its own design, and these sensors installed on both legs, right left hip and right left shin. So we have 4 sensors that strongly synchronized each other and collect this data into the one hub in smartphone, and then processed for calculating general parameters that I explained in further. For calculating these parameters, we use orthogonal coordinates, like on this slide, stationarity coordinate connected with Earth, and local coordinates that connected to each sensor. And these equations well known, but we use for this equation, like calculating angles from data from accelerometer and gyroscope, in air modeling, calculating trajectories of airplane. So it's not new. So it's this equations well-known equations. So, but what new? We use different angles between direction of each sensors, and further I show that it's very important part of calculation and very important information about characteristics of moving. Here you can see how we mount the sensor on the legs. And here experiments. We set 3 types experiments. First is normal walking, and second when dysfunction one of leg, right leg limping or left leg limping. It's need for describe how change these parameters during limping or normal, for compare these parameters. And here the statistical average value of this parameter for each angles, alpha, beta, gamma, low hip, right hip, low shin, and right shin. So 4 sensors for measuring these angles. I'm sorry. Unexpected call. Comparative analysis, normal walking and limping, show that on a limping we have change average value. It's real. It's understanding for this changing. So walking characteristic has changed. So average value has changed too. So, mean distortion showed us almost equal. So we don't see any big changes. Any big changes are in RMSD. Despite this, when we use differential angles, we have this is curves of normal walking and limping left leg. You can see changes in the form of curves, and then very interesting result. We have strongly changing in not only average value but in the RMSD. It showed us that each segments have different angles between vectors of direction, top segment and bottom segment, and this changing in the time give us with different type of walking, give us very interesting information about how this person walking. And this information can be used for categorization of type of walking and segments of walking. And finally, we calculate very interesting graphs. It's a mutual trajectories of angles. Here is dependencies between left shin, same angles, left shin and left hip, left shin and right shin. So top, bottom, and cross. Right shin, right hip, left hip, right hip. In a normal, you can see very interesting trajectory with almost same repeated. We're walking, we repeat movement very good. So when person limping, these trajectories don't have any repeating. So it's just I don't know.
Chaotic.
I don't know.
No, it's not chaotic, that it's collapsed from the loop. It's just
so. Moreover, these graphs get us possibilities to make an visual feedback during person check walking by itself. So we can construct this biofeedback when person look on her own trajectory, and when this trajectory not normal, correct himself, herself. Yeah. So this very interesting result, and we going to use this result for design these apps for personal using of these patients or using with biomedical assessment in rehabilitation procedures. And conclusions. So I collect all parts that I said, usually we can open the collaboration. You can see our email, me, and you can check the QR code and send me an email letter. Thank you for attention. Question, please.
Speaking about the software you are using, which programming language are you using?
Python?
Sorry?
Python?
Python.
Yep. So it's all calculation and all models being constructed in the Python language. Modeling with using Mykhailo, a little bit GPT, like helper, for doing some
just coding assistant.
Yeah.
Michael, what is with these gray lines on your course in the previous slide?
I see.
Yeah, because you have the colored one and the gray one. What the gray one means?
This is color curves is mean curves after this plane interpolation. And the gray curves, it's raw data. So it's just little bit normalized.
Okay. I mean, because this very, very interesting results, because if you look on the left bottom pictures, the red line, I mean, there's a right shin versus right hip. And if you look, okay, normal pattern of normal walking, it's like some picture, like this, I don't know, eggplant shape, But in limping, this mean line is almost oval, but raw data have some, I don't know, some lemniscata type, this like butterfly-like picture, and probably it's like, it's a curve or deformed to the previous one, and actually that is what you mentioned. I think that is an absolutely fantastic thing, because if you look at this gray line and perhaps advertising how the person should limping his leg in different way, perhaps from an energical and muscles work we could propose rehabilitation modification to the pattern, how it would change, try to be more left one than the right one, I mean. Because I think this deformation here is just like it could be visualized completely. I mean, about these red curves, how left and right are different. So I'm just thinking on. It's not a question, okay.
No any rules for these curves when limping. So spline interpolation give us some average average data, average summarizing. Moreover, I have my bed in this picture. I'm sorry, but I want to focus you that scale on these graphs is different. If you can see here's 40 minus 40 degrees, and these 10.
Yeah.
Here the same, 60, almost 60, 50, and minus 20, or 15, 50 degrees, and here only minus 5. So just amplitude has changed. Not only curve, but if I construct these curves in same
Scale.
scale, sorry, same scale, so it's could be different in, like this is bigger and this only in there inside. So not only form of curves, but absolute value of this. Moreover, when I return to I return to differential angles, I mark in red this one. Keep angles. Here, but near the zero. But there, it's more bigger. So what you can, result, what we can think about this? When one leg have immobility, our how is it calls?
Hip joint.
So we correct, making correction immobility because of very strange moving of middle of our body. So just like moving with very big changing angles in medium or mid of body. So it's very big pressure on the
Vertebral column.
our musculoskeletal system. So that's all. Thank you. Next one. S5-1.7, exploring maternal placental fetal interactions, hybrid modeling approach for biomedical engineering. Just a second. One second. Please.
Okay, my name is Viorel and I will be presenting in a way innovative model called here hybrid model for a quite simple problem, in a way simple, but it's not simple at all. Let me start. First, speaking about modeling. Here we have a list of some modeling approaches like equation-based model, statistical model, especially statistical model quite common in research, and the last two are not so common. I mean system dynamics modeling and agent-based modeling, hereafter used as hybrid modeling, their combination. Speaking about the problem, speaking about adults, we have many possibilities to monitor the conditions and processes that are running in the human body, including, in our case, pregnancy. But speaking about the fetus is quite difficult, and the only one possibility of monitoring is continuously registering the heartbeats of of the fetus. Here I will be speaking about oxygen delivery. Everyone knows that oxygen is vital for our life, and even short periods of time with less than necessary oxygen or hypoxia can cause irreversible changes, including in the fetus. And the idea is to an attempt to solve this lack of information concerning the fetus. This is the general idea. Now, I'm presenting here this modeling approach, ADM plus SDM, as a potential solution. And what I will be presenting was performed in NetLogo environment, including the programming language with the same name, NetLogo. Here we have a part of the overall picture. In fact, here we have the placenta. Placenta as that part that connects the fetus body with the mother body. Here is a magnification. We have two systems, circulatory system of the mother and of the fetus that will continue in the umbilical cord to the fetus, delivering the oxygen I'm speaking about. Now, speaking about system dynamics part of the model, on the screen you can see three subsystems. Subsystem number one is the cardiovascular system of the mother. Subsystem number two, or system number two, in fact, is the same for the fetus. And three is the connection between them, or placenta, presented in that small diagram or image over there. Now, speaking about, before I'm changing, what we have here. Here we have such structure as squares representing stalks. We have arrows representing the flow, and we have many diamonds representing different parameters. And all these arrows connecting small size arrows or thin arrows representing relation. And this relation can be described mathematically. And next slide is about this. Here are main elements mathematical formulas for this model. Number one is the general equation for volume change. Number two is for pressure in vessels, aorta or vena cava, main vessels in the human body. Ventricular pressure is a different story because here it happens in completely different way than in the vessels. And you can see this long formula. What is important here is this part. Time varying elastance that is changing during the cardiac cycle. And here is the formula, is an exponential function. Here the time is the time of a cardiac cycle. And we are deducting this systolic part. Just speaking about some details, I do not know if I should do this or not, but just in case. And finally, how can we calculate the oxygen delivery? DO2 stands for oxygen delivery. CO means cardiac output, or the amount of blood ejected by the heart during 1 minute. 134 is a constant showing the amount of oxygen 1 gram of hemoglobin can bind. And finally, we have the saturation of the oxygen saturation of the arterial blood. This is the final formula the model is using to simulate and show the amount of oxygen that is delivered to the fetus that can not be otherwise measured. Next, here we have the graphical interface of the model. The agent-based part is here. Here we have these red circles are erythrocytes. This is the maternal side. We have here a membrane, and here we have the fetal side. And these blue color circles representing oxygen. and a darker color, almost black color, carbon dioxide. They are traveling across this membrane in the opposite way. What else we have here? Here we have a scenario chooser. This model let the user use 6 scenarios. I will be explaining later. Here we have the running parameters for the maternal part, cardiovascular parameters, pressures and volumes. Here the same for the fetal. This monitor is presenting the running or overtime, how they are changing in the mother. Here on the fetus, but a little bit later. Below we have a number of sliders that let us to adjust different parameters. First of all, the cardiovascular function, including the heart rate here, as well as the hemoglobin, because the hemoglobin can be low in the mother and then originally will have problems. And the saturation that again can be adjusted to the case we are dealing with. On the right side, this window shows oxygen delivery. Here we have the oxygen delivered in milliliters per minute to the fetus. And you can notice here a red color line representing the hypoxia limit. A normal situation is when this upper part of the green area is above this line. And on this slide we have the normal situation. Below we have a number of sliders, 6 sliders. Again, over the 5, here we have 6. We can adjust different parameters concerning placenta and the fetus, including the fetal heart rate that is being measured. Next, speaking about what happens during delivery or childbirth in regular terms. The same green area, the same red line, and you can see that during uterine contraction it will get lowered, but will not cross the red line, meaning that everything is okay. On these monitors we can see different parameters, including the amount of oxygen delivered to the fetus during 1 minute. Here we have some pathological conditions. First one is, well, most of them pathological. The first one is normal scenario, when there is no contraction. You can see that the upper limit is above the red line and it's okay. Next shows a situation when there is maternal anemia is present. You can see that it's close, but periodically it will get lower than this hypoxia red line. Here we have complex pathology and uterine contraction, denoted by this notch. Originally it was lower. If we have here 84, here we have 74 milliliters per minute. And here we have the same, but under general anesthesia. General anesthesia will influence all these problems, and meaning that if a woman during labor and delivery are some problems, they will get worse during anesthesia. Next, I would like to show you how it looks in reality. All this, it's a video of the model interface. You can see how erythrocytes moving, how curves changing, and you can see that in fact this is the scenario of complex pathology without contraction. Anyway, and now contraction, you can see that it gets even much lower than the hypoxia limit. Okay, it's short. I will let it finish. So this is the idea. The hemoglobin, for instance, is 110, is lower than normal. Next, there is quite a serious condition under the name placental abruption or abruptio placenta. This is in Latin. Here we have the fetus, surrounded by the uterus. Here we have placenta, and something happened, and this placenta get disconnected from the uterus all. This will present with external bleeding if it happens here, and or internal bleeding that is quite difficult to detect. But anyway, the fetus will suffer. And one more reason why such a model can help. And here we have how it looks like on the model. You can see hypoxia limit. This is abruptio placenta in 2 steps. And you can see it gets lower and lower and lower until it will can reach the 0 line, meaning no oxygen delivered to the fetus. This was the visual part. Now, shortly, the research part. The idea is that we can use a complex of or a number of parameters that are registered or monitored in a regular intensive care unit, in our case, obstetrical intensive care unit, including the fetus, the only one from the fetus. And by combining this and applying the Kolmogorov-Chaitin complexity on the matrices, numerical matrices with the value of the parameters, we can use this to detect the event of placental abruption. And here the red line denotes, in fact, is the same we have seen on the upper limit of the green area on the previous slide. Here is something calculated based on other parameters, not calculating the amount of oxygen delivered. And you can see, here is the impact time. You can see the changes at this level also changing. In this way, there seems to be a possibility of using indirect parameters to detect the event. So this, in fact, is one of the main goals of this model. Not counting as a tool for students to better understand what is happening and play with different parameters and see how it will look like. The final parameters of interest, oxygen delivery to the fetus. Now conclusions. It seems that this model effectively simulates oxygen delivery to the fetus. We may speak about a dual purpose as educational tool, as a clinical prototype. And evidently, as a clinical prototype, it will need refinement and validation on real-time data. This work is part of a larger research, postdoctoral research program, Intelligent Software Methods and Algorithms for Intensive Care, complexity science approach, implemented at the Technical University of Moldova. And that's all. Here is the address. This model can be downloaded, and if someone likes, can play with. Evidently, you will need to install NetLogo, but it's not a problem. It's open source.
Yeah. So?
Yes.
Oh, thank you very much. Question for presenter, please.
Yeah, I have some. Could you please show a math model?
Well, it's not, it's some elements.
Yeah.
Because the number of equation, you can imagine the number of diamonds on the previous scheme is over 30 equations. Here are the main equations.
Yeah, but I don't really understand. So the main parameter that you computed in your model is this last one, yeah? Oxygen delivery parameter,
yes.
Yeah? And here is the CaO2. What does this mean? It's saturation, yeah? It's oxygen saturation.
This one? Yeah, oxygen saturation. And this is a regular formula, it's not my formula.
Yeah, okay.
A regular formula for calculating the...
And HB?
Hemoglobin.
That is also, it's like it's a parameter that we should put in this slide bar.
Yes, these are the parameters used to estimate the amount of oxygen delivered, not necessarily in the fetus, in every human.
And CO?
Cardiac output, or the amount of blood ejected by the heart during 1 minute. Again, is something not invented for this.
My question, I don't really understand. In this model, all these parameters just set up in the model.
No.
No?
No, of course not. Hemoglobin is set up. Saturation you can play with in some cases. But cardiac output that is based on all this formula, not. You can simulate some conditions. This is about cardiovascular system of the mother or of the fetus. You can set some, but the final result will be cardiac output.
So then this is not a parameter. The CO is a function of time?
No. CO means the amount of blood ejected by the heart during 1 minute. Normal range is between 4 and 8 liters in a normal individual. With some problems, it can get lower. And one of the cases I presented there, I probably...
I'm just not understand what happens in your graphs, because in your simulation there is like some dependence, that's all the time, and or what about this?
Yes, everything here is over time. This one and here also is over time.
That is also the time.
Is a dynamic, system dynamic model. This is why it's system dynamic.
And CO of these parameters is a function of time.
That is computed over time, but is not a parameter of time. It's over time based on volumes. Let's go to the formula here. Volume. In fact, cardiac output is a volume over time, a volume ejected. But for calculating this, here we need the pressure and we also need the resistance parameters. Is the limit between two cardiac segments. For instance, mitral valve in the left part of the heart between the left atrium and left ventricle, or between here we have left ventricle and aorta. There is another bottleneck, let's say this way, and we have some resistance, and this will influence the volume. But what happens, and this, in fact, this part is a continuous part, but heart is not working continuously. It's ejecting and then stops, ejecting, stops. And this long formula is about this. And one of the main elements is this time varying elastance of the myocardium.
But it's just Gauss formula.
Sorry?
It's just a Gauss.
Well, I think it resembles Gauss formula, just the elements are medical.
And this is time is just the same as the t value in equation 1. It's the same time.
This is simply the duration of a cardiac cycle. For instance, 60 beats per minute will mean that it will be 1 second duration of cardiac cycle. And we deduct the systolic component because there is diastole and systole. And we deduct the systolic component, and this will give us this time varying elastance that in fact in this formula is used to demonstrate the pulsatile nature of the blood flow at this level.
Okay, and the last one, if I may. In these pictures then you show us this, I don't know how to say this, animation of the pictures, then this oxygen and CO2 molecules are moving. What is that and how is it modeled?
Just one for me.
Just
this I'm never skim slide.
Oh,
can you
change slide?
Okay, thank you.
yeah, this one,
Yes.
this red circles, blue one, black one.
This is the ABM part. Everything else is the system SDM part, or system dynamics part of the model.
Okay.
Model.
It's like in a Simulink in MATLAB. You just put this
Yes,
integrators and create
you
actually
can
some
do
different
many
don't know it. Okay.
programming languages.
Yeah, okay.
This
okay.
is simple because the interface is quite nice, let's put it this way, and
Yeah.
is easy to understand. And this part represent the movement of blood first, two parts. In placenta there are two parts, maternal part and fetal part, divided by a membrane. And through this membrane, the gases in our case, and not only gases, nutrients for instance, will travel from the mother to the fetus, and carbon dioxide from the fetus to the mother. And this simply represents how, just a visual
But what about the laws?
Sorry?
What about the physics? Which law you presented here? How membrane filtering or pumping? What happens here from physical sense?
Yes, is
I'm not conceptual, but which
I don't know, it's Mykhail's Menten transporter. If something tells you about this, again is a concept about the diffusion through the membrane. I did not present it here because it would be quite too complex.
Okay. Diffusion model.
Diffusion model.
Now it's almost there. Okay. Thank you.
By the way, it's few words. By the way, calculating of cardio output is not easy task.
Well,
Because it's just simplifying model of calculating cardio output.
well, speaking about cardiac output, in a clinical setting is indeed
Surgical.
a very difficult task.
Yep.
And needs quite specific equipment.
Yep.
And there are different methods, invasive, non-invasive, etc. Here we speak about simulation. How close is the simulation to the reality or to the parameters measured by this measure, thermodilutional, etc. This is another question. But anyway, preliminary checking is, this is a simulation, let's put this way, it's a simulation with the potential to be aligned with real patient data in the future. It's a
By the way-
prototype.
By the way, your work is not focusing on the cardio output.
No, this is
you're
one
focusing
parameter.
on the another
Absolutely,
task.
absolutely.
Yep.
Although here we have cardiac output, and if we follow, you will see that it will be more or less constant.
Yep.
We have, but what really counts here is this amount of oxygen delivered to placenta, over 100 milliliters in the mother, and what gets to the fetus is slightly over 84.
I agree. Yep.
These are two parameters important in our case, but they are based on many other parameters important for
But no law.
that might be important for other cases.
Real situation.
Thank you. Any questions?
Any question? Yeah, bro.
Thank you. Part S5 1.8 towards more protected medical data, assessing the security of web and email infrastructures in SMEs in the Republic of Moldova.
Good afternoon, dear colleagues. My name is Alexey Arina, and I'm from Technical University of Moldova. Together with my colleague, we study how secure data that are manipulated by medical, small and medium enterprises from Republic of Moldova. The results show important gaps between best practices and real-world implementation. Let's see. Doesn't work.
Ключи.
Can change.
Just click.
Change.
Just
Okay. In Moldova, healthcare become more and more digital. That is very easy and commode for healthcare domain, but is more risky too. When patient data moves online through websites of medical CME or through email systems. They become very important and interesting attack vectors for attackers because medical data are very sensitive data. So in 2024, an accident or an incident was in a network of laboratories from Moldova when thousands of test results were shown online. And not just for this network, but for a laboratory in Germany and Romania too, who works with this network. That's why we wanted to see how secure are our CME at all about web and email security. So we used a methodology with open source intelligent gathering tools and OSINT approach. Thus, we didn't attack any systems. We just collected publicly available data. And we have some security metrics like TLS certificate analysis to see how secure they transmit their data. DNS and email security through DNSSEC and other email parameters for security. Security headers that are small web rules that can set in the browsers. And result processing with combined results for comparison. Okay. So you have analyzed 80 domains and subdomains from 20 CMEs in Moldova. What you see here is that 65% have valid TLS configurations, but they use not very strong security configurations for TLS, like cryptology chiffres. 35% have outdated or weak TLS protocols like TLS 1.0 and TLS 1.1, and just 50% have hash H STS, that is a header that had to protect against SSL stripping attacks when we think that we access a secure domain, but in our browsers we done SSL strip and we use just HTTP and transfer data unencrypted. And one site have DNSSEC enabled. DNSSEC is a parameter that permit anti-typo squatting attacks. And it's against malware or unlegitimate versions of medical sites. Next, we checked for web vulnerabilities and security headers. So only one domain from 20 CMEs received grade B, but it don't have a security headers properly configured. 45% scores D, that is very low, and half of medical domains don't have any headers, security headers at all. You can see here most frequent missing headers that are very important for secure communications, like HSTS, content security policy, X-frame options, and referral policy. And only 61% of domains has used secure cookies, and they are proper configured. Then you check for email security and authentication. In check TLS assessment, you can see that you have just half of email subdomains properly secured email transmission, valid and have valid TLS certificates. 25% have self-signed certificates that do not assure security, and 25% have not valid certificate or encryptions or of their data. Very important for email security is SPF, the KIM and the MARC attributes, because SPF can decide which domain can send an email. The KIM that this email is trusted or is authentic, and the MARC what action will be done if something goes wrong. And the primary domain implement in 50% of cases SPF, the KIM or the MARC, but just in 25% of SPF, they are used in subdomains. And they are missing authentication protocol in some domains that is vulnerable to spoofing or phishing attacks, even when the primary domain appear secure. So it's not enough to secure just domains, but on subdomains too. We have 3 research questions about security adequacy, strengths in configuration, and weaknesses. And in my conclusion for first one was that we have partial compliance with TLS, DNS SECs, and we have serious gaps in best practice adoption. We need for a measurable way to assess maturity. For the second, strengths in configuration, some of the domains use TLS 1.3, that is the most secure version of this protocol, and secure cookies. But we need improvement and scoring. And the last one, we have missing security headers, email attributes like the KIM, the MARC, and weak hardening. And we need for a unified comparable monitoring model that we propose, security compliance score. So you have proposed this model. Rationale for this was derived from research question 1 to 3. In this structure, we have 6 elements or indicators, like TLS configuration, security headers, DNSSEC implementation, secure cookies, email authentication, and server exposure. Like an outcome, we have a quantifiable model and comparable security score for CME. each of these 6 parameters scored from 0 to 100 and weighted by real world impact of them. In my findings, we assess 80 medical CME domains and subdomains, revealed gaps between practices and actual implementations, measured vulnerabilities with TLS and DNSSEC misconfiguration, missing security headers and absent email authentication. These weaknesses expose sensitive healthcare data to significant risks and calling for systematic improvement. Like a research impact is awareness, highlights real-world misconfiguration in their security implication, and methodological contribution through a replicable quantitative model that can be used to assess web and email security of a CME. And like a future work, we want to develop automated remediation and monitoring tools, conduct longitudinal studies to track the evolution of CME security practices, and expand the model applicability to regional and cross-sectional sector context. Thank you.
Thank you. Question, please, the presenter.
Interesting, but quite specific for me.
Yes.
But anyway, I will ask one question. Who should address the problem with this, the existing situation? Who is?
I think that both IT experts who implement security configuration, but healthcare personnel need awareness to understand that medical data are interesting for everyone, and it's just this leak data that was in 2024, it's not a cyber attack, it's just an improper configuration of its IT system. I think that it's both, from the both parts.
I have one question. Tell me please, what is suggest to improve this problem? A problem with not good security status of for medical medical sites of medical databases. What you should do for now?
For now is awareness belong medical staff that they need to understand they don't have IT expert every day, and they need to understand how this data, how sensitive is this data, and what can because they can impact human lives. Because a change in a test or in a results can conduct to very serious problems. I think that awareness is the first stage.
I know this problem is a real complex problem. It's a physical layer of communication, is a logical layer, is data set communication, database communication, and cloud communication security. So
Yes.
it's a huge numbers of tasks. So it's this problem inside the country, or
It's
inside.
not just inside.
It's a private clinical.
We assess just private clinical because they send the results through email They can access the results, the patients can access the results from internet on their web platform here in Moldova. And it's very complex, and we need a very granular approach to this problem. But to protect data in transit was the first meaning of this article.
Okay, thank you. Professor Artur?
Yeah, I have two questions. The first one, technical one. Could you please move to the previous slide? Even more. Sorry, yeah, exactly. That's SCTS parameter. That's like a matrix that estimates the efficiency, or I don't know, the level of cybersecurity of the system. Yeah? Am I right?
Yes,
okay.
yes.
And the W sub I is a weighted parameters. Yeah?
Yes.
Who, how, and in which way estimates these values, and what about the what are they actually?
We can estimate this impact just from the impact of this parameter. Yes, TLS configuration, for example, in this is the more important. Why? Because data in transit, how we access, how we authenticate on this site are the main security poster that you can assess. Or for example, DNSSEC implementation. Yes, you have in simple words Google.com. Right? You have now more than 1,000 sites that belong to Google.com and not are Google.com. And DNSSEC assure that when we access Google.com, this is for their servers. Yes.
That name server. It's server could translate IP
Server
address
who
to name.
deliver this service for customer, for clients. You can do this weight just on impact that they have on their data.
Yeah, that is clear for me that I could do that, but I'm not a cyber security specialist, so I cannot set them. Who set them and in which way? So this sum of all these W parameters actually should equal to 1. Yeah?
Yes, equal to 1, and they are different like weight. And we can set just on international reports and impacts of data.
Okay, and what about S sub I? You mentioned that it's all from 0 to 100. That is somehow calculated through, I don't know, through this automatic system of controlling, or it's also just set by a group of experts?
Maybe for just a group of experts, but at national level, not at CME's level, yes, because they need to do research to find TLS configuration, is data confidentiality, DNSSEC implementation, is confidentiality of domain. We need to know that data protection is first of all data confidentiality in medical data and data integrity. Yes, but because very important. And based on this and of results of international reports, you can set a weight impact, yes, of each of these.
And this data integrity you mentioned, that's very interesting parameter. Is it from your study, from Moldova? I think in Ukraine the same stuff. Is it at risk at the moment through this IT system? Is it possible that data integrity could be broken under some non-human based mistake, but from the system?
Yes, from the system, because email authentication, yes, we have SPM, the KIM, the mark. The KIM is an attribute that is not configured in any system of CME, of Moldovan CME, but they are this attribute can confirm through digital signature that this email is from who say it is. Yes, from, I don't know, in vitro MD. Yes? This attribute is very important because he showed that this message is authenticated, is authentic or not.
Okay, thank you very much.
Thank you.
More question?
Hey.
No question. Thank you very much.
Thank you.
Benefit of from AI and these benefits and this new good afternoon everyone. Today I would like to present our research on artificial intelligence in healthcare, where we looked at real-world integration as well as the emerging cybersecurity risks and challenges. We will go over the background, why we chose this research topic, which methodology we use, and then into the results that we found, so the real-world applications, the risks, regulatory landscape, and we will conclude with recommendations as well as way forward. So why AI and healthcare? We all see a large hype nowadays around artificial intelligence, as it offers a large number of opportunities, especially when it comes to analyzing large amounts of data. Healthcare is a sector or domain which can benefit from AI, and these benefits and this new implementation and innovation can improve the health in our society. So our research is a continuation of a previous research in 2023, where we looked initially at the theoretical level on the risks, especially on the cybersecurity risks. However, in the last 2 years, we have seen AI going from theory to permanent use. And on the right side, you can see just a small number of implementations that nowadays are in real systems and in healthcare. We use a selective literature review. Our source was Springer. We chose articles in the last 2 years, between May 2023 and May 2025. We focused on articles available openly, and we screened diagonally a number of articles against real-world AI adoption, infrastructure, cybersecurity reviews, and we selected a number of articles from the ones found that we believe to be the most promising. So, among the main results that we have identified during this selective research are that AI is used, and in many cases we see it being used in convergence with other technologies such as blockchain, for example, for the electronic health records. We see a lot of solutions integrated in our clinical workflows, and these have clear advantages such as operational efficiency or reductions in clerical workload. And many articles also highlighted already the risks and concerns such as inconsistencies in the data accuracies or how dependent does one become from AI-generated content. It was also interesting to see that ethics has been a topic widely discussed. So we can say that ethics is crucial, and it's complementary to the cybersecurity area when it needs to come to evaluating AI implementations. We had also seen that a lot of these implementations have been evaluated using the so-called technology acceptance model, where interviewees were asked on their perceived usefulness of innovation and the perceived ease of use. So we see that AI overall has large interest from the respondees, and also many already understand the potential risks, ethical questions or concerns in terms of this new innovation. However, when it comes to innovation, we also need to look on the positive side. So, for example, in 2024, FDA in the United States authorized 169 medical devices that use AI. And from these, 125 were solely in radiology. Now, there can be a number of reasons why this area is number one in this. However, it's clear that when you have data stored in electronic format, makes it much more easier to use it with AI, especially when you can train certain models, especially when you can train them in a very trustworthy manner, and it can help you a lot. Now, as part of other benefits, of course, you can see that the triage can be done faster, workflows can be accelerated. However, again, challenges posed by AI per se need to be tackled, such as how do you handle false positives, or when you use the wrong data in, you need sometimes to do follow-up evaluations and so on. Other areas are also identified in the research, such as the use in diabetics and chronic care. When you look at tailored education, for example, you can do any training in any language for any audience. You can use AI in research and development, where you can see where some drugs can be used for other purposes and how can this increase the precision. You can embed AI with telemedicine, which has been in our area for many years already. And lately, last but not the least, AI should support and not replace in any way clinicians. Now, if we talk about risks, we have cybersecurity risks. We can, in theory, divide them between generic IT risks, so risks that are specific to an IT system, to a computer. Unfortunately, the maturity in this healthcare sector is still emerging, still needs to grow. And we see the same main issues such as weak, for example, network segmentation, reliance on outdated systems, security practices, and it's very hard to actually add these security controls after, because it's much more efficient when these are done at the design phase. Nonetheless, we need to look at AI-related risks. So, first one that directly impacts IT systems is that cyber attack nowadays can happen much more faster using AI, and it's not a matter of minutes. And, for example, you can have a lot of very sophisticated phishing attacks. But when it comes to AI technology per se or concept, you need to look at data bias. Was data poisoned in any way that was used to train the model? Can it be manipulated? Do you see actually the prompts for how this model and tool has been built using AI? What about regulation? Is regulation at the national level in place? Does it actually help, or one needs to look more at the regional level? And of course, any new AI implementation, especially when done in a silo approach, so individually, can increase the so-called shadow IT, so IT systems outside the purview of IT. In the regulatory area, we can see that there are some developments, and these are significant at the regional level. We also see 2 standards from ISO already published. We see another standard in progress. And of course, the regulatory landscape will set the tone on how this new technology and concept can be developed. In terms of recommendations, well, after reviewing the paper, it was nice to see that cybersecurity risks, ethical concerns are already tackled at the research level, which is good. We want to also recommend again and again security by design, but as well ethics and privacy by design, to ensure that the basic controls and concepts are included in the design of any system, including AI. And if we look at cybersecurity controls, we can have the technical ones, we can have operations, and here, besides the usual IT best practices, we need to look at how do we handle operations and monitor operations about AI. Do we know when the model is hallucinating or giving outputs that do not exist? Or the model is drifting, so starts to work or do other tasks that we was not asked to do? How do we respond to such incidents? At the governance level as well, we need to see how do we actually document an AI asset. Do we look at the so-called bill of materials? Do we document everything that we know about it? How do we do governance of such systems? How do we align it to standards? And then, of course, how are the above technical and operational controls implemented? And last but not least, of course, continue raising awareness as security culture is a main element in all this discussion. In conclusion, AI is accelerating, especially in healthcare. Risks are evolving. However, we also need to understand that many innovation can have benefits. And we also note that the main aim needs to remain a secure, ethical, and effective patient care by taking into account all these aspects and making sure that we have good governance and we have good cybersecurity controls embedded. Thank you very much for your attention.
Okay.
Good afternoon, everyone.
So, no, all in our list has done.
No questions for the man?
There's no questions for movie.
We will listen to you if you have.
So, I want to say thank you all, and for good presentation, for discuss, and good luck. Thank you.
Thank you.